Hello, my name is Braxton Shaw, and today I'll be starting this presentation off about open heart action potentials. Myself, Ethan Larson, Ethan Lovejoy, and Sterling Murdoch all participated in this study, and will each be presenting to you briefly on kind of what we did and what we found during this study, and what our findings mean to the world today. So in the past decade, there's been a huge boom in knowledge about the human heart, particularly about the human heart during open heart surgery. For our experiment, we set out to discover the effects of open heart surgery on the action potential of cardiac muscle fibers. Previous similar studies have been conducted on non-human mammalian heart muscles, specifically canine hearts, rabbit hearts, and various other mammal hearts, but similar studies on human heart muscles have not been conducted thus far. In our study, we will conduct similar experiments to those done on mammalian hearts on human cardiac muscle to understand how similar human heart muscle fibers react to action potential stimulus compared to the previous findings done on non-human mammalian cardiac muscles. For our study, we obtained atrial tissue from 5 different individuals. The ages of these participants were 2, 10, 28, 32, and 53. All patients were awaiting open heart surgery. The patient aged 28 had aortic incompetence, while the patient aged 53 had aortic stenosis. All other patients had atrial septal defects. A visual of this can be seen on the left-hand side of the screen, where there is a defect in the septum between the 2 atria, and the left atrium is leaking blood into the right atrium. We also obtained ventricular tissue from 2 individuals. The ages of these participants were 4 and 6. Both participants had ventricular septal defects. So during our experiment, we went through a few different stages and steps to get our results. So during surgery, pieces of pectinate muscle were excised from the aforementioned patients. The pieces were taken from the right atrium or trabeculae carnae of the right ventricle. Immediately after, the tissues were immersed in Tyrode solution and taken from the OR to a laboratory. An image of the Tyrode solution can be seen in the bottom right corner of the screen. Muscle strips were then fixed in a tissue chamber with constant flow of Tyrode solution. This fixation was done within 30 minutes of excision in all cases. The solution was equilibrated with gas, providing a pH between 7.2 and 7.4. CO2 and O2 were used as the primary buffers. Various experiments were done by injecting different chemicals into the tissue bath. Chemicals included acetylcholine, chloride, epinephrine, and hydrochloride. We'll get into more details about these individual experiments later on in the video. Each having contact with the preparation for only a few seconds. Membrane potentials were measured using glass micropipettes filled with 3 molar potassium chloride. The recording system consisted of a cathode with negative capacitance and a Tektronix twin beam type 502 oscilloscope. This machine can be seen on the left-hand side of the screen.
Okay, now on to the results. So our first results we have here, we show figure 1, which shows action potential of a single ventricular fiber. We measured a rapid upstroke that was followed by a plateau in the region of a positive potential. So after this plateau, the resting membrane potential of the muscle fiber would repolarize to the RMP. We measured the action potentials of the muscle fiber 24 different times and found that the average RMP would always return back to negative 87 millivolts. So we found that very interesting with these results. For our next figure 2, showing here the upstrokes of action potentials. So with these results, these were recorded simultaneously with 2 intracellular electrodes impaled at a distance of 2.5 millimeters in a ventricular muscle fiber. I have an example with the picture on the right here of some electrodes on heart muscle. And you can see kind of just how the electrical impulses are sent through the heart muscle, giving us upstrokes of action potentials, which were recorded in figure 2. We did find that when we increased the extracellular concentration of potassium, the upstroke velocity in the action potentials was reduced. We also found that during our testing, we determined the conductance velocity to be 1.3 meters per second. There was some variation in the data, but it wasn't related to the distance of the electrodes in the muscle fiber. As we were measuring action potentials, we didn't find any action potentials with prominent spikes that is typical of the Purkinje system. So I have a heart here, and you can see the Purkinje fibers at the bottom of the screen. I highlighted them with circles. With the Purkinje system, it's found in the interventricular walls of the heart, just beneath the endocardium. These fibers are specialized myocardial fibers that conduct an electrical stimulus or impulse that helps the heart to, it synchronizes contractions, so it can help the heart beat. It's essential for maintaining a consistent heart rhythm. And with these findings, we found that conductance velocity to be higher than those measured in dog ventricle. When we compared the two conductance, we found that human hearts, the conductance velocity is higher. So that was pretty interesting. My colleague Ethan Larson will present on the rest of our findings of this experiment.
Like my good friend and colleague Braxton Straw said, during this experiment, we took tissue samples from the ventricular heart and we placed it into a solution. The solution is called a thyroid solution. Something important to know about a thyroid solution is that it has specific concentrations of certain ions. One thing we really wanted to test was the effects of changing the extracellular concentration of sodium on the action potential of a heart. So if you look at the screen, on the left side of the screen, you'll see a thyroid solution. A thyroid solution has 153 millimoles of sodium. First thing we did is we put the heart tissue into this thyroid solution, and after a while, we placed it to a second solution. We'll call it solution B. Solution B only had 16 millimoles of sodium. So on this figure 3A, we can see the effects of putting the heart in the thyroid solution and testing the action potential there, and then putting in solution B and testing it again. We did this at 2 different times. We tested it at 7 minutes after placing the new solution and after 10. As you see on the graph, you'll see a blue line pointing to one line. That's the action potential when it was in the thyroid solution. As you can notice, the action potential was long, right? After being 7 minutes in the solution B, you can see with that purple line, the action potential got a lot shorter. And then after 10 minutes, it got even shorter. Something important to note is that we had to increase the strength of the stimulus when we put into solution B. So the stimulus had to be stronger when we decreased the concentration of sodium. Figure 3B shows the upstroke. As you can see, the upstroke in the thyroid solution when we had a high extracellular concentration of sodium was very fast. That slope is almost vertical. But if you look at solution B, you can see that the slope is a lot longer. It takes a lot longer to hit the top. So we noticed that 2 things from this experiment, that the extracellular concentration of sodium is very important for the duration of action potentials and the upstroke. Something we actually really noticed too that really compared our studies to other findings in the past is that when we put the ventricular muscle or ventricular tissue into solution B, the effects decrease as we can see from the graph. But after putting it back into the thyroid solution, it quickly recovered. This has been seen in mammal tissue in the past. So it kind of furthered our conclusion that there's some correlation between mammalian hearts and human hearts. The next thing we wanted to do was to see the effects of the rate of stimulus on the duration of action potentials. So to show this, we had fast stimulus rates and slow stimulus rates. Representing fast stimulus rates in this picture is a pronghorn antelope, the fastest animal in North America, and slow stimulus rates is a tortoise, which is famously slow, right? So figure 4, this is showing how the effects of the rate of the stimulus affects the duration of the action potentials. So first you'll see that red line on the very far side of the graph. That's when it was at 24 per minute. As you can see, the action potential is very long, right? Right next to it, you'll see a brown line pointing to, kind of looks like the same line, but it actually isn't. That was at 42 per minute. So something we learned is between 24 and 42 being the rate of stimulus, there really wasn't that big of a change. When we increased the rate of stimulus to 60, though, the action potential decreased dramatically. And we kept on doing this until we got to 162. And as you look at 162, you can see that the length of the action potential is a lot slower. So something we learned is that as we increase the rate of the stimulus, the time of our action potential, how quick they are going to get progressively slower, or progressively faster, sorry. I will now turn over the time to Ethan Lovejoy for our conclusion and talk about how this experiment can really impact the future and what we're really hopeful for.
All right, so let's talk about why our research is important. As my colleagues pointed out earlier, a lot of the research that's been done on hearts hasn't been done on human hearts at all. It's been done on animal hearts, and that's because human hearts are a lot more valuable, right, than animal hearts, than animals just in general. And as a result of that, we know a lot about animal hearts. We don't know a whole lot about human hearts. Our research, though, is the first one where we've been able to take human heart muscle and take it into a laboratory setting and test it with various ions and all these different things that we haven't been able to do before. The extent to the research before this has really only been on animal hearts or putting electrodes on human hearts to try and figure out how the action potentials are working and figure out what's going on. Our research had concurrent findings with Weidman, Draper, and Maya 2, especially when talking about the upstroke and conduction velocity of the heart. We had concurrent findings talking about fiber radius and the velocity of action potentials, the same findings that Katz found. And Brady and Woodbury with extracellular sodium and heart muscles, they had all done this research on animal hearts, though. And so this is really great that we're able to find these same things on human hearts. And this is important because we can experiment on animal hearts and find out more about hearts, and we know that the same findings will apply to human hearts. So this is really fascinating, and it's really great because as the world of medicine progresses and we develop newer and more complicated surgeries, especially with the heart, we need to be able to test those and make sure that they're even viable in the first place using animals. They also serve as a great way for surgeons and medical students to practice these really complicated surgeries. If they can do it on animal hearts, then they can do it on human hearts. And we can develop new technologies like breathing machines and machines that will take over the function of the heart when we need to do something major to the heart. We've also been thinking about what would happen if we wanted to take out a heart from one person and put it in someone else, do heart transplants between humans or even between animals. If we're similar enough, we may be able to use animal hearts and substitute them for humans whose hearts are broken or damaged or diseased. So this is really incredible research that we're doing, and it's super exciting to see where this is going to go in the future. We're really excited for it. We're grateful that you were able to listen to our presentation. Here are sources here, references. If you'd like to read our paper, you can click on that link at the very bottom. It'll take you to the Circulation Research Journal, and you can read kind of more of what we did, get a little bit more in depth into it. And we're grateful that you listened to us, and thank you so much. Have a good day.